All right, so we'll start out with dilution basics here. So what am I talking about when I'm talking about making a dilution? So what you're simply doing is reducing the concentration of a solution by adding water, solvent, or another diluent to it. So used in microbiology, we reduce the number of organisms during sample preparation. So if we're looking at the picture down below, on the far left, you have the most concentrated solution. In micro terms, you would expect to have the highest amount of microorganisms in the far left side. And as you go towards the right, the solution becomes more dilute and you would expect less microorganisms as you have a more diluted solution. So dilution basics, what are we trying to achieve with doing dilutions in microplating? So we want our plates that we read to be within a certain countable range. So why is there a countable range and why is it important? We need the most statistically accurate result that's going to be the most meaningful for you. So if you don't do a dilution, you might have a plate that's covered in bacteria that would be too numerous to count. And having a result that's too numerous to count won't exactly be helpful for you to use that as tracking and trending, or it won't give you a meaningful result for your product. So adding these dilutions in is going to give you the best accurate result. So how do we go about doing that? We're going to perform a controlled reduction of bacteria so that we can choose the best plates to interpret that fit within that countable range. And we'll get into countable ranges a little bit later in the webinar. So what's the problem? So first, when we have our plating and we're ready to set up, we don't know the starting point. We don't know the bacterial load of the environment. If you're doing an environmental sponge or your product, you can't just look at a piece of cheese and say, it's probably got about 10,000 CFU bacteria, I should do an extra dilution. So that's the problem. You can't just look at it and know when you're going to need it and when you're not going to need those extra dilutions. So the solution for that is analyzing that product, that environmental sample at different concentrations within the series. So doing a multitude of different dilutions. So when you're going to start, you're going to want to prepare a 1 to 10 dilution typically. So what that means is it's 1 part of your original sample in 10 parts total. So if I break this down, 1 part of your sample and 9 parts diluent, which makes up 10 parts total. So the first step of your sample preparation, like I said, is typically preparing that initial 1 to 10 dilution. So that can mean 10 grams of cheese into 90 mils of diluent, 11 mils of milk into 99 mils of diluent, so on and so forth. So your first step is that 1 to 10 dilution. And then your second step beyond that is going to be the serial dilutions. So if we look at this picture here, on the top left corner, you're going to start with your original inoculum. You'll take 1 mil from there into 9 mils of broth, and that would yield you a 1 to 10 dilution. So if you plate 1 mil from that first tube, you're going to have what looks like a big cluster and a big mess, and you can't see individual colonies down on that plating auger below. So you would need an additional dilution. So you take 1 mil from that 1 to 10 dilution into another 9 mil tube of broth, and that yields you a 1 to 100 dilution, which looks a little bit better on the auger plate, but still not anywhere near being able to do an accurate count. So you'll do it again, take 1 mil from your 1 to 100 dilution and yield a 1 to 1000 dilution, plate 1 mil, and it's getting pretty good, but we still want to separate those colonies out a little bit further. So we'll do another dilution set, 1 to 10,000. Plate 1 mil, and that has a very good bacterial count. All those individual colonies, they're very easy to see. You don't have those big clusters or those big groups of colonies. So that's an ideal plate to count. And just did 1 further than that, and you're able to see that further reduction in bacteria in the 1 to 100,000 dilution. Okay. So if we were looking here to calculate a result, I would use the 1 to 10,000 plate here. So on the bottom, it says for calculation, you're going to take the number of colonies on the plate times the reciprocal dilution factor. It's going to be your number of bacteria per mil. So if we use that 1 to 10,000 plate, if you've got 32 colonies on a plate from your 1 to 10,000 dilution, then your count is going to be 32, which is your number of colonies, times that denominator, the 1 to 10,000 dilution. So 32 times 10,000 is going to yield 320,000 bacteria or CFU per mil in that sample. Okay. We had a question come in. Someone had asked, is serial dilution something you sort of play with to find the ideal dilution? And it's always better to start out with more dilutions than you think you need, and then kind of get an idea of what's going to work best for you going forward. So it is something that you'll need to play with, but make sure that you do more dilutions is better than less, so you don't have to go back and reset something up again. Okay. So this picture here is a little bit more of a realistic, real world example here. We have some salmonella growth on chromogenic agar. So you'll see with the 10 to the 3 dilution, we've got an absolute mess of bacteria growth, nothing you could count. And as you dilute it further and further, the 10 to the 4, the 10 to the 5th, you're getting some better differentiation in colonies, but you don't really see that best plate until you hit the 10 to the 6th dilution and then the 10 to the 7th. And that's where you can really start to see individual colony types and use those to get an accurate count of your sample. So we have this picture that's a 10 to the 6th, and you could calculate your result by using the count of bacteria, 114 count, multiplied by your dilution, the 10 to the 6th. But what we're not seeing in this picture is how much was plated onto the agar plate. So if I take it back one step, you can see we plated 1 mil from there. And then we were able to calculate what our dilution factor was. However, in here, we don't know if we plated 1 mil. We don't have this information. It's missing. So we're not able to accurately say that what our dilution factor was, because you can't automatically assume it's 10 to the 6th because it's based on the amount that you plated. So let's talk a little bit more about dilution factors. So if you start with a sample of E. coli, you're going to make that initial 1 to 10 dilution. You'll take 1 mil, put it into 9 mils. You've got a 1 to 10, and so on and so forth. Your dilution factor is going to change based on the amount that you take out of that dilution and plate. So your dilution factor is calculated by the reciprocal of the dilution multiplied by the amount plated. So let's just take that 1 to 10 dilution, for example. So your dilution factor is going to be the reciprocal of the dilution. So it's just going to be 10, that denominator of the dilution. And then you're going to multiply it by 1 mil, because that's how much you plated. So your dilution factor is going to be 10. So if you're always taking out a mil, it's going to be very easy, very straightforward to know what your dilution factor is. Where it changes is if you're plating something other than 1 mil. Because if you're taking that, let's say, for example, the very last one, the 1 to 1 million dilution that yields you 24 CFU on the plate. Instead of plating 1 mil, what if we plated 0.1 mil? So then you'd be taking that 1 million times 0.1, which is going to make your dilution factor now 100,000. So then you take 24 times 100,000, and that would yield you your result. So it's important to always consider the dilution factor and not just go with what the dilution is, because it matters the amount that you put onto that plate. All right. So talking serial dilutions, what can go wrong here? So if your sample was mixed poorly, if your bacteria is not evenly distributed or clumps up, that can cause issues with plate interpretation, okay? Your inaccurate measurements could also cause some issue here with serial dilutions. If your technicians have poor pipetting technique, where you aspirate too quickly, you just release that plunger, you might aspirate some air. Or if you have the wrong tip immersion, that can also cause you to aspirate air bubbles and not get you to get that full volume that you're looking for. Another thing for inconsistent measurements is having inconsistent volumes in your dilution blanks. So in those tubes that we were putting 1 mil into, and we're supposed to contain 9 mils of diluent, it can really throw off your dilution series if your tubes sometimes have either higher volumes or lower volumes. If you're using one that has 8 mils instead of 9 or 10 mils instead of 9. You see this a lot with labs that make their own dilution blanks. There can be little inconsistencies there. But just make sure if you're using these dilution blanks that you have 9 mils. Something else that can also play into this is if you lose focus and you forgot what was done, what wasn't done, if you skipped or double inoculated a tube, you know, somebody came in and said, hey, I need your help with this, and you just kind of got drawn away from where you were in the process. It's easy to just come back and say, I think I inoculated that, or I don't think I did, and double inoculate, and then all of a sudden your dilution series doesn't make sense anymore. You can also have mislabeling of plates corresponding to the dilution factor. You know, so if you think you prepared a 1 to 100 dilution, but it was really your 1 to 1000 dilution, that can obviously throw things off as well. And just because one of those major things that can go wrong is pipetting, I just wanted to touch on a couple points of proper pipetting techniques. You want to make sure that you grab a pipette that's in the proper range for the volume that you need. You want to verify that the pipette is set at the correct volume, even if it's typically set at a constant value and doesn't really ever change. It's still good to check in case the dial was accidentally adjusted during cleaning. I know that can sometimes happen. So it's always good to get in the habit of checking your pipette set at the correct volume. During use, you want to push the plunger down to that first mechanical stop every time. Don't go all the way down. Just make sure you hit that first stop. It's also a good idea to use consistent pipetting rhythm. For pipetting large volumes like 1 mil or greater, it's important to pause after you have the sample pickup just to allow the pipette to fully aspirate the sample before you remove the tip. Just like a 1 second pause is good enough. That way it takes the time needed to draw up that larger volume, and you're not taking the pipette out and then it would aspirate air for the rest of the sample. Okay. So sample prep matters. So when plates are difficult to read, results can be inaccurate and you're not really getting a good idea of what's in your sample. So some of the things that can happen is you've got sticky cells. This can be a result of insufficient mixing, shaking, vortexing during sample prep. So make sure you're not rushing these steps, that you're taking the time to accurately mix it and shake it and vortex each one of your dilutions as you go. You can have an uneven distribution on a plate or a Petrifilm. I've seen accidents happen where the wrong spreader is used for the wrong Petrifilm. I've seen where the sample itself is outside the boundaries for the Petrifilm. You know, someone will press the spreader on and the sample will just leak right off the sides. You know, that's not going to give you a good accurate count. Another thing is swarming cells. If you take too much time between plating and mixing with your auger, if you're doing a pour plate, or spreading it, if you're doing spread plates, that can cause a very large clump of cells in the center of your auger plate. So you want to make sure that you don't take too much time in between plating and mixing or spreading. Another thing is if you have too much liquid on a spread plate, that can also cause swarming cells or grouping of cells. And it'd be very difficult to get an accurate CFU count off of that. Also, you have some bacteria that are just more likely to swarm. They have a flagella, which means they're motile bacteria. So some of them might clump together. So plate count results are reported in CFU. So they are called colony forming units. They can be given as a number of CFU per mil for a liquid sample, CFU per gram for a product sample, or CFU per sampled area for a sponge. So ideally it's formed from a single cell if the sample is prepared correctly. If you've done all those steps to prevent sticky cells, uneven distribution on your plate, and avoiding swarming cells, So if you've done everything correctly, ideally, your colony forming unit would stem from one single cell. And this is critical to the accuracy and reproducibility of your test. Okay. So your plating tests are quantitative methods. You use these when you want to figure out how much is there. It's good to use for indicator organisms, coliform, EB, things like that. The portion of the sample is prepared and then incubated, and the result that you get out from that, like I said on the previous page, is numerical, reported in a CFU per gram per mil per sampled area. For example, 150 CFU per gram. Okay. And we've got a couple different plate count methods here. I'll briefly go over a pour plate, a spread plate. For the Petrifilm, last year we did a Petrifilm use and interpretation webinar. So if you want more about specific information to Petrifilm, go back and view that on our website. But I'm going to go over pour and spread plate here a little bit further. So looking at the pictures that are up on the slide right now, you'll notice a difference between the top one and the bottom one, where the top one is a spread plate. You can see that the colonies are just sitting on top of the agar, whereas the picture on the bottom, you can see better that they're kind of evenly dispersed throughout the agar. So the top one is a spread plate and the bottom one is a pour plate. So we'll go through those in greater detail. Okay. So on the left side here, we have a pour plate. So if you have an empty Petri dish, then you'll take your sample, 1 mil or 0.1 mil sometimes, and put it in the empty Petri dish. And you'll have your melted nutrient agar, whatever you're using for your enumeration. You'll pour it in there, and then you're going to swirl that around to mix. And what that is going to do is mix your agar with the amount of sample that you pipetted. You're going to want to swirl it clockwise, counterclockwise, a little bit of forward and backward, as well as left and right motion, just to make sure that that is evenly distributed throughout that agar plate. And with this method, you're going to have colonies that are growing in and on top of the solidified medium. Versus on the right-hand side, where we have a spread plate method, the agar is already solidified in the plate. You're going to take your 0.1 mil of your sample, place it on top, and then you'll use an inoculum spreader to spread that solution over the agar evenly. So I've seen people use disposable spreaders. People have glass ones, metal ones that are autoclavable. All those work fine, whatever you prefer. Just make sure that the inoculum is spread evenly over the surface, that you don't have a large amount pooling in one area, okay? And then the colonies on that plate will only grow on the surface of the medium. So let's talk a little bit about advantages and disadvantages for each method. So the pour plate, you're able to plate a larger sample volume. You've got some increased sensitivity there. You could plate 1 mil versus with the spread plate, it's recommended to do 0.1 mil. So you have a little bit more buffering, neutralization of the sample with that agar pour plate. And those motile colonies that I said previously for swarming cells, you know, those bacteria that have flagella like to swarm, you'll have less issues using a pour plate because they're not able to spread as much on the surface, being distributed throughout the pour plate. As far as a spread plate, it's much easier to see different colony morphology with the growth that just is on the surface of the plate. You'll have more oxygen present for aerobic organisms. It's very helpful for opaque augers. If you're familiar with Baird-Parker, it's a very opaque tan color and would be hard to see through if you set that up as a pour plate. So those are typically done as spread plates. The thing with spread plates is you need to have a dry surface. So those plates should be prepared in advance. You should have a dry surface for plating. If you have any moisture on there, you're going to get clumping, sticky cells, and it's not going to yield a very good interpretation, a very good distinct colony forming unit. So you'll have a lot of those colonies kind of meld together if you have moisture on the plate. So it's always good to prepare those in advance and have them dried out. Another good thing about the spread plate is that you can purchase these, and they're a little bit easier for people rather than going through the hassle of melting and pouring your own plates. So we'll go through some colony counting tips. It's very helpful to have a dark field colony counter. It's pictured over there on the right-hand side. So it's backlit. There's a light bulb in there. There's also a grid that you can see. It's very helpful for counting auger plates. And then there's also a magnification glass over the top that gives you a 10x magnification. So when you're doing these plates, you're going to want to follow those colony counting rules. We'll go over those as well as the countable ranges. Another good thing is to know how to interpret difficult plates when you've got spreaders, when you've got confluent growth, when you've got TNTC. Make sure you and your techs know how to interpret those things, which we'll go over here as well. Okay, so for countable ranges, they're going to vary depending upon your plate count method. Countable ranges for a Petri dish versus a Petri film versus something like a membrane filtration, they're all going to be different based on your plate count method. It's also dependent upon the type and size of bacteria you're looking for. So it's going to be different for a coliform versus an APC count. So why do these countable ranges matter? Why do we have them? So counts outside of the specified countable range might give you an erroneous indication of the actual bacterial composition of the sample. So for example, if we've got a countable range of 25 to 250, dilution factors might exaggerate those low counts. Those counts that are less than 25 might be a little bit exaggerated. And if you've got a crowded plate that's greater than 250, it might be difficult to count, and it might inhibit the growth of some bacteria, resulting in a low count and not as accurate count. So if you need to report outside of that countable range, if we stick with this example, less than 25 or more than 250 colonies, you would report that as an estimated count using the asterisk symbol. So you might see that on your C of A's as we report out an estimated 240 CFU per gram. So if we took that from a 10 dilution factor plate and we had 24 colonies, you'd have the 24 colonies, obviously less than the 25. So you'd want to use that estimated symbol and apply your dilution factor. So you would get an estimated 240 CFU per gram. Okay. So digging in a little bit more on these countable ranges, there are defined upper limits, like I said, based on bacteria colony size and behavior. Coliform is going to be different than a staph. It's going to be different than an APC. So this is also based on the surface of the plate. Like I said, a Petri film countable range is going to be different than an auger plate countable range. So this upper limit is needed because these colonies will eventually compete for space and nutrients. So the assumption at the upper limit, if you look at figure 1, the estimated versus observed plate counts, in red is what you're expecting, and then in blue is what you're actually observing and seeing. And as the chart goes on, you see that you don't get the expected count. It kind of tapers off there. So eventually that observed CFU will fall off at the expected number at some point. And eventually this difference between the expected and the observed becomes too significant and makes the count inaccurate. Like I said, those bacteria will eventually compete for space and nutrients, and there's a limited amount in that Petri film or in that auger plate. So it could end up giving you a misrepresentation. So that's why there are defined upper limits. So then why are there defined lower limits? I get this question a lot. It is based on the limit of detection. So in figure 2 here, if you're looking at the error of estimate of low plate counts, you're going to get more poor accuracy when using plate counts less than 25. So as the CFU plate count drops below the countable range, below that 25 CFU, the percent error increases rapidly. And this is just a limit of detection thing. So if it's above the upper limit, we'll use an estimated sign. And if it's below the lower limit, we'll use an estimated sign for these reasons. So plates within the countable range should be used to determine your final results. You'll take the number of colonies and multiply that by the dilution factor to obtain your final result. So for example, you have 127 colonies on a plate and your dilution factor is 10. You multiply those together, you get 1,270. Final results should be reported to two significant figures using whole numbers. So rounded to sig figs using the example above. If you have 1,270, you're going to round up to 1,300. And that rounding is always your final step. So as I said before, these results are reported CFU per gram per mil per sampled area. If your plates are done in duplicate, you want to count the plates within the countable range and then average counts to obtain your final colony count. So after your average is determined, the result is then rounded to 2 sig figs. Like I said, the rounding is always the last step. So let's go through an example here. So if you have 2 plates that you're going to average, they're both 10 dilution factors. One plate you have 126 colonies, the other plate you have 122. We're going to take that average, which is going to give us an average of 124 colonies. You apply your dilution factor, which is 10, which will give you 1,240. And then lastly, you'll apply that rounding rule and you'll round down and your final result would be 1,200 CFU per gram. Okay, we'll go through some additional examples here. Keep in mind, like I said earlier, plates below or above that countable range are going to use that estimated symbol, the asterisk there. So for example, we have an APC pour plate. That countable range is 25 to 250. So the amount of bacteria on there is 15 CFU. Your dilution factor is 10. So you're gonna take the 15, multiply by your 10, and you're gonna have an estimated 150 CFU per gram. And we have an estimated sign there because 15 CFU is less than 25, okay? We take another one, for example, 272 colonies, which is greater than our upper limit of 250. Multiply that by our dilution factor, and you're gonna get an estimated 27,000 CFU per gram, okay? So it's one example below the limit and one example above the limit where we use estimated signs in both, okay? So if your plates have no colonies, no growth at all, then your final result would be estimated less than your lowest dilution factor. We'll stick with that APC pour plate, countable range 25 to 250 CFU. Like I said, you don't have any growth, your colony count is 0, your lowest dilution factor is 10. So your final result is an estimated less than 10 CFU per gram. Very similarly, if your count is still 0 and your lowest dilution factor that you set up is 100, then your final result is going to be an estimated less than 100 CFU per gram. So if there are no plates within countable range, you're going to want to use the plate with the count closest to the upper end of the countable range. So why is this? Your accuracy is going to be better at the higher end of the range than at the lower end of the range. So another way is to say this is you're going to use that one towards the upper end of the countable range because that sample's been manipulated less, diluted less, so there's less chance for error, okay? Your APC pour plate countable range, again, 25 to 250. On your 1 to 10 dilution, you have 268, which is above your countable range. On your 1 to 100, you have 21, which is below your countable range. You're going to want to use that 1 to 10 dilution that's above your upper limit because that sample has been diluted less. There's less chance for error. You know, there's less chance for technician error, for pipetting error, all of those other factors that we talked about. You're going to want to use that at the upper end of the range to calculate your result. Okay. So because they're both out of your countable range, you're still going to use the estimated sign. And you take the 268, multiply by your dilution factor, and then round up. So you'll have 2,700 CFU per gram, and estimated. Let's talk a little bit about plate interpretation and some things that can be difficult. So one of those things is particulate matter. A a lot of samples have particulate matter in it, and it can be difficult for technicians, and they could possibly mistake particles or undissolved medium as pinpoint colonies. I've seen it many, many times in the lab. We're fortunate enough here to have an instrument called a stereo zoom, which helps us zoom in on that, and we're able to see colony morphology. If you look at the pictures down below, you can see that bacteria, like on the right, it's the yeast colony. They're typically smooth, round or oblong, and very uniform in shape. However, these particulates that you can see around it, and more so in the picture on the left, are kind of crystalline looking. They're jagged, they're rough, textured in appearance. And sometimes if you're unable to decide and the foreign matter versus pinpoint colonies isn't clear to you, a gram stain is definitely a good way to check it out and make sure that you're counting only bacteria and not mistaking that for any kind of undissolved particulate matter. Another plate interpretation situation is if you have a spreader. Spreading colonies can be chains, clumps, clusters of colonies that aren't distinctly separate. They can be films that develop sometimes underneath that auger if you've got a pour plate, or films that develop around the edge on the surface. You can see in the picture on the right, sorry, my apologies, the picture on the right there, picture of a spreader. So you want to select a plate without spreader colonies if possible. If you can't use a plate without one, then you can estimate the plate by using the area that's unaffected by the spreader if possible. For plates that are covered in spreader growth and you don't have anywhere to count, your result may be best utilized as a spreader result. Okay. Don't try to count individual colonies within a spreader. This is probably going to lead to inaccurate counts. If only one chain or clump exists, it's typically counted as a single colony. If your chain appears to originate from separate sources, you can tell either by like one's a different color, one's a different texture. So if they appear to be different, count each source as one colony. Okay. Let's touch a little bit on Tntc plates. So the abbreviation Tntc means too numerous to count. So this abbreviation is used when there are too many colonies present on the plate to count. So a Tntc result can be used when plates have more colonies than that upper limit of the countable range. But you're also able to make estimated counts on plates when there are distinct separated colonies. So if I'm looking at the plate on the top, I would report that out as a Tntc result. There's no way that I could get an accurate estimation using that plate. There's just too much growth present. However, the one on the bottom is probably slightly over the upper range for my countable limit. I could make a good estimation off of that plate just because you can see distinct separate colonies there. Okay. So if all your plates that you set up are Tntc, it's a good idea to retest with higher dilutions if possible. But like I said, you can use an estimate. So to estimate a plate, you want to determine the average number of colonies per square. And this is where having those colony counters that I talked about before comes in handy because it has that grid plate on the back, which is going to help you do your estimating. So you'll find the average number of colonies per square. And then you'll multiply the average number of colonies per square by your total area of the plate, which will give you an estimated number of colonies per plate. And then you'll multiply that number by your dilution factor to get your estimated final result. Okay. So when I'm talking about average per square, and I'm talking about the colony counter that I was referencing before, this grid plate is very helpful when you're doing an estimation. So if you have less than 10 colonies per square, you should count 12 colonies perpendicular to one another, highlighted in yellow here. So if you do this, you want to make sure that you don't count this center one twice. Okay, so you've got 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. So if you're counting it across this way, don't count it again when you come down and estimate this way. Only count this center one once. If you have more than 10 colonies per square, then you're going to want to count 4. So where these green ones are highlighted here, just make sure that the 4 you choose are representative of your plate. So I don't want to choose 4 with the most colony growth in them. I don't want to choose 4 with the least amount in them. So make sure you choose 4 that are representative for your plate so you're getting an accurate count. Okay. Another plate situation you could run into is confluent growth. So on some auger plates, confluent growth can be overlooked. It can be missed because sometimes it doesn't look like defined colony growth. The plate on the right looks pretty uniform, but is actually a layer covering the plate. In this case, it's E. coli, and it can't be distinguished as either a layer that resulted from one colony that spread, or it's a large volume of colonies that grew together into a layer. So that's why we don't call it a spreader. We don't call it TNTC, it is confluent growth, okay? So is it TNTC, is it confluent growth, or is it a spreader? So your technicians should take care to note the difference between these. So the difference between them is TNTC is distinguished by a defined amount of colonies specified more than your countable upper limit range. So it's too numerous to count, but you can see defined colonies there. It's not that layer of growth that we just talked about. Spreaders can be chains, clumps of bacteria, and the confluent growth that we just went over defined as a layer that's covering your plate. Okay. So how do you keep your counts accurate between techs? So make sure that your techs read and review colony counting rules and that you all agree on how to interpret plates. So all your techs should be able to read and interpret plates with reproducible and comparable results. It's a good recommendation to implement a verification to ensure accuracy between your techs and make sure you're all on the same page as far as how to read and interpret. So you have each tech count the same set of plate 2 times. So preferably you have an hour in between each read. What this is going to do is make sure that that tech has reproducible counts. When they read it first, walk away, an hour later, come back and read it and still get a similar result. You're also going to do this with all of your techs. So you're going to take that text read and compare it to all of your other texts within your group. So there are a couple of different references that talk about this. The SMEDP says that a technician should be within 8% of their own counts when they do their 2 counts, and within 10% of your group of technicians. So the compendium then says technicians should be within 5% of yourself, a little bit of tighter restriction there, and then 10% within the group. So here at Cherney, we do follow the compendium, and we make sure that our technicians do this quarterly and have 5% within their self reads and then 10% within the group of technicians to make sure that everybody here is on the same page for how to read and interpret different colony counts. Technician counting verification where inaccurate counts can be caused by analyst fatigue, carelessness, impaired vision. You can have the issue of mistaking particles and undissolved medium, like I said earlier. Oil droplets can be difficult to interpret on pour plates. Some certain precipitates or air bubbles, they can be difficult. So make sure that if you have somebody who has inaccurate counts, that you're taking the time to address this with them and say, you know, make sure that you're not reading this as pinpoint colonies or misinterpreting it in any way. Corrective action for out of range results. You're going to want to review the colony counting methods, make sure that all the texts are following the same reporting rules as far as when to use estimates, when to average, how to round to sig figs. You want to make sure that everybody's on the same page for that. Review the method specific to the test and make sure that the technician is counting the correct colony types. Certain petrifilms, they can be difficult. You have to know exactly what type of growth you're looking for, what color changes you're looking for. So make sure that your techs are aware and counting the correct colonies only. If you're still having issues, it could be helpful to use a hand tally or a Sharpie to mark the colony counted and to prevent missing or double counting any colonies. It could be necessary to adjust work schedules to prevent analyst fatigue, and just take a closer look there into any other issues that might be causing those inaccurate results.